Well, hello everyone, and thank you for participating in today's event, Innovative Solutions to Address Impacts of Ocean Acidification. My name is Jessie Turner, and I'm the project manager for the OA Alliance, and very happy to be co-hosting this event today with partners from Ocean Foundation and from Peace Boat. And this event, of course, is in part a way to celebrate World Oceans Day and the programming this week that has followed. It is in part to share innovative solutions that governments and civil society are helping to advance that support the implementation of UN SDG 14. And it is in part a way to keep up ambition for urgent climate action to reduce carbon emissions. And before we begin, I do want to take an important moment this afternoon to acknowledge the pain and frustration that people are feeling all over the world, and particularly here in the United States. The last few months and weeks have continued to bring a steady stream of difficult and painful news, impacts to our collective global health, and urgent discourse about the racism and systemic inequities prevalent within our policies, our communities, and across civic life. And it is a profound and important moment that we are experiencing together. And it is indeed upon all of us to take actions in our professional and our personal lives. And the Alliance is an entity that largely exists to information share and to elevate messages. And we must always do better and be conscientious and consider whose information is being shared and whose messages are being elevated. And finally, I want to acknowledge that climate change and its most damaging impacts are a direct result of colonialism and the commoditization of our world and the humans that share it. And therefore, when we're thinking about climate change, we must always consider it and have it seen through the lens of climate justice and racial justice. And in my mind, that is a moral imperative. So thank you for being here today and for all that you do to advance this shared work. It really means a lot. I know it's been a long week for folks. And so it really is special to have your participation here today. It's not lost on me. This is a choice to show up and be here. So for those of you that are new to the Alliance as members or as partners, I'll just begin with a quick overview of our work before introducing today's agenda and lineup of speakers. The OA Alliance, as you heard, brings together governments and organizations from across the globe dedicated to taking urgent action to protect coastal communities and livelihoods from OA and other climate ocean impacts. And so together we're working to elevate urgency and ambition for climate action, to integrate ocean into climate commitments, and to translate knowledge into policy action. And since our launch, we've grown to over 100 members, and we're still growing. It's great that we have such a strong mix of national, state, municipal, sovereign, tribal, Indigenous and First Nation governments, along with many dedicated affiliate partners like NGOs, industry leaders, and local academia. The Alliance, how we roll is, we call for the creation of OA action plans, which are meant to describe actions that members are taking or will take to better understand and respond to the threat of ocean acidification and other climate ocean impacts within their regions. And specifically, we focus on commitments is to focus on things that will reduce carbon dioxide emissions, the #1 cause of ocean acidification, advance scientific understanding, reduce local pollutions that exacerbate OA, protect the environment and coastal communities from impacts of a changing ocean. And this particularly alludes to the adaptation and resiliency strategies that you'll hear about today. Expand public awareness, and then of course, sustain international and multi-governmental support for addressing this global problem. And we keep in mind that not all OA action plans will have the same framework or structure, as there's really no one-size-fits-all approach for governments in thinking through how they're going to uniquely experience or respond to this issue. That said, we do have a framework and we do have a toolkit which provides a little bit more prescriptive recommendations and showcases examples from example actions and examples from members. And I just want to call your attention that we have updated this toolkit recently, and it's now available on our website, in addition to more guidance about how to get started. And just to give you a sense of how we run through some of these topics and examples, here's Action 2, Advanced Scientific Understanding. We have various categories of actions that governments or affiliate members might consider, followed by pages that have examples from members, links to their work, just to give you a little bit more hands-on look at what this entails and looks like over our membership. So again, today's World Ocean Day theme is Innovation for Sustainable Ocean. And I'm happy to introduce our speakers for today and an excellent program that will focus on innovative actions that are helping local communities and regions around the world explore shellfish hatchery techniques like buffering, selecting for tolerance, and building resilience, deploying nature-based solutions such as submerged aquatic vegetation, which you'll hear about today, develop predictive forecasting models and local monitoring that can inform adaptation responses, and then driving this private and public collaboration that will help us advance knowledge, and you'll hear a little bit more about that from the end of our speakers set today. So a great lineup, and just to give you all a sense, we've got, I did schedule this for an hour and a half. We'll probably wrap a little bit early. All of our speakers will have about 8 to 10 minutes to present. So we'll just give you that heads up in advance and appreciate your attention sticking with us. First, we'll have Dr. Cliff Law with the National Institute of Water and Atmospheric Research in New Zealand, followed by Dr. Nicole Price with Bigelow Laboratories for Ocean Sciences in Maine. And then we'll have Jacqueline Ramsey with the Alutiiq Pride Shellfish Hatchery in Alaska, followed by Alexis Valori Orton with Ocean Foundation, Dr. Wiley Evans with Hakai Institute, and Miss Emily McGlone with Peace Boat to close us out, and one of our co-hosts here today who's welcomed us to be part of this UN Oceans Week programming. So thank you, Emily. All right, so with that, let's see. I'll go ahead and bring up Dr. Oh, I'll go ahead and bring up Cliff's presentation. Oh no. Okay. Oh no. We can do this. All right. All right, Cliff.
My name is Cliff Law. I'm a marine scientist working for the National Institute of Water and Atmospheric Research and the University of Otago in New Zealand. And I'm also the lead scientist on New Zealand's Ocean Acidification Program, which is known as CARIM, Coastal Acidification Rates, Impact and Management, which I'll be telling you about in this presentation. The motivation behind this study is the ocean around New Zealand is acidifying. We're well aware of this from two complementary time series that have been carried out, one in the atmosphere, shown in red, of the atmospheric carbon dioxide, which has been stepping up over the last 40 years, and one from the water side, where measurements in the surface ocean in subantarctic waters off Otago is showing an increase in the blue triangles there, which of surface ocean carbon dioxide that parallels the increase in the atmosphere. At the bottom of this plot, you can see that there's a complementary decrease in the pH of the surface ocean over that same 20-year period. So we have cause and effect there in terms of atmospheric CO2 going into the ocean and the dropping in pH. And that drop in pH is something around the scale of around 7% increase in the acidity over the 20 years. So with that in mind, about 5 years ago, we brought over experts from the US and also shellfish farmers from the US who are already experiencing issues on the US West Coast in the oyster hatcheries. And this raised a red flag in New Zealand that we really need to be focusing on the issue of coastal acidification, particularly as this might affect some of our major uses of the coast of New Zealand. So this led to us developing a national ocean acidification impacts program with a focus across the country, 3 different sites. These 3 sites are shown in your map there. They're all different in terms of their morphology, also about the level of urban or anthropogenic influence in these environments, but they're also home to 3 ecologically and also economically important species, and they are the fish snapper, the green-lipped mussel, and the black-footed abalone, or POA as it's known. And so the question about our program was, how will coastal species, ecosystems and economies be affected by ocean acidification? So here's the structure of the CARIN project. Largely, we're focused initially around the rates. So there we get information by monitoring at these 3 sites. And we also use this information to develop projections of how pH will change in the future at these locations. We then take this information and that informs our impact studies on our 3 key species. And here we look at different aspects of those species. For example, with the snapper, we just tend to look at the larva, particularly around recruitment and behavior. Whereas with the shellfish, we look at different aspects such as settlement, physiology, energetics, and shell formation. And an important aspect of this is not just looking at the direct impacts of acidification, but also the indirect impacts by, for example, food supply in terms of primary production, and also the quantity and quality, and also about substrate as well for the settlement of larvae. So we take all this impacts information and we feed that through into population models for each of these species to establish what, or get some indication of what their future success will be. And then this information informs the approaches to deal with this and therefore the management for these species. So just to give you some more information on different aspects of the program, this is the National Monitoring Network, NZOAON, of which CARIM is part of. And NZOAON consists of 15 national sites that have been run for about 5 years, where we have different stakeholders. And there's a whole range here from everything from local communities, local councils, ministries, fisheries, and the shellfish industry, all collecting water samples for us that are processed at a central facility down on the South Island. And so we get information on all these sites on the variation in pH, and this allows us to establish baseline conditions, the national variability, and also to identify vulnerable sites. And of course, this information is now we focus on particular sectoral region, and particularly the case of the Firth of Thames, the region in the north of the North Island, where, as the central figure shows, we see a real decline in pH as you move into the Firth itself, as you get nearer the coast. From the offshore waters, you can see that there's already experiences pH levels of around 7.9, which is really equivalent to what the open ocean is predicted to experience by the middle of this century. So in other words, these coastal waters are already ahead of the game in terms of acidification. So we gain information on this by monitoring both in the central waters, but also on mussel farms in the more coastal regions. We have local engagement in terms of collecting water samples. We put out public fact sheets to keep local stakeholders informed, and also develop projections of how pH will change in the future, which are specifically tailored to these sites. In terms of the impacts on key species, just to give you a summary of some of the results here, on the left side you can see the snapper larvae, and so under high CO2 we do see some effects on their auditory senses, certainly their swimming ability and also their metabolism. But the good news is that generally we see an increase in survival rates for snapper larvae under high CO2. Now the poa don't fare so well. Now adults, they're at high CO2, it tends to reduce their spawning success. For juveniles, they have a reduced growth and survival, and their shell integrity is also compromised. But it's the larvae that are the real weak point. They, under pH, which is projected for the end of this century, they show much slower development and a much higher rate of abnormalities than present day larvae show. So clearly this is suggesting that maybe this is the larvae are the weak spot in the future success of them. And this kind of similar response is confirmed in the green lip mussel. Now, whereas generally the life stages are relatively resilient to low pH, we see some impacts on gonad development in adults and reduced survival in the juveniles. It's really the embryos, particularly the first 48 hours of the embryos, that are particularly vulnerable to low pH. And so once again, this emphasizes the fact that it's a full life history assessment is necessary to establish really where the bottleneck to survival are within a species. So what are we going to do about this? Well, clearly one solution we can have is actually looking at the factors that are driving coastal acidification. And to do this, we've focused on the Firth of Thames, and we've used coupled physical dynamic models for these regions, which allow us to look at how sensitive the pH is in the water to the different factors that drive it. So, for example, we know that pH in the Firth is really quite low in autumn, and this is due to the modification of the biogeochemistry of the water by the runoff that's coming of the catchment. And so the model tends to back up the observations that suggest that if we can manage the nutrients and the ocean organic carbon that's being input from the land into the Firth, then we can actually potentially reduce the impacts of coastal acidification. Now, another solution that focuses more closely on one of the coastal economies, and that's the shellfish aquaculture industry. And so we've worked with them to try out different approaches. Now, one of these is just straightforward monitoring, really trying to understand the variability in pH in the carbonate system around mussel farms themselves, but also potentially some solutions to future-proofing them. So one way to do this potentially is that we have a lot of waste shell lying around New Zealand, and this is all carbonate. So if we can get that carbonate back into solution around a mussel farm, we can provide some sort of buffering against low pH, at least on a farm scale. Similarly, there's also aeration that can be used. So, for example, if we strategically aerate at night, when there's excess carbon dioxide in the water, or under warm periods, for example, we can drive out that excess and therefore protect the mussels when they are basically most vulnerable. Finally, in terms of the green-net mussel industry, there's the potential for resilience and adaptation within different families. So some experiments have been carried out looking at using the stock from New Zealand's aquaculture selective breeding program. The adults have been exposed to low pH, and then the response of the early larval stage have been assessed in terms of their survival and their shell length. And this the initial indications are from this data set that it suggests there is a capacity to adapt, that some families can actually show more resistance to low pH than others. And this is useful information to the aquaculture industry because it shows that they could potentially focus on these more resilient strains in their future applications. So just to summarize there, I'll just finish off by saying that, that it's important, some recommendations from this project is that you should really take a national perspective. It's important to not only understand the variability in your country, but also the fact that where those sensitive regions are. It's important to engage stakeholders. Clearly, all coastal stakeholders have a stake in the future of their coastal waters, and the more information you provide, the more they're engaged, the more value there becomes from the research. It's important, once again, to get a full life history understanding of the species that you're interested in, and also focus on things that both have, an economic as well as an ecological value. And finally, I would say that we know a lot about impacts of ocean acidification now, and now is the critical time to focus on tools and solutions to that. So with that, I'll leave you with the acknowledgements to a number of people who've contributed to the CARIN project, and thank you for your attention.
Fantastic. Thank you so much, Cliff, for that presentation out of New Zealand with NIWA, an amazing partner of the OA Alliance. And let me just double check on participants. I apologize, I'm leaving people waiting outside the waiting room, so I want to make sure I'm getting folks in here. All right. Nicole, can you hear me okay?
Yes, I can.
Great. So we'll go ahead and queue up our next presentation from Dr. Nicole Price with Bigelow Laboratory for Ocean Sciences in Maine, who will be talking about kelp farming as a strategy for remediating OA and improving shellfish cultivation, which you'll hear about directly. All right. All right, Nicole, take it away.
Thanks, Jessie. Well, thank you, Jessie and Emily, for inviting me to speak today. This is a wonderful set of talks that you've put together. And I want to start by acknowledging that this is the result of a 4-year collaboration across a couple of academic institutions, and to echo what Cliff's just said, stakeholders. We've engaged directly with kelp farmers and mussel farmers to be involved in this project. So the Island Institute, Bigelow Lab for Ocean Sciences, the University of New Hampshire have been really leading this project, but with collaborations from Atlantic Sea Farms, Ocean Approved, and Bangs Island Mussels. Thanks. Next slide, please. So it started as a pilot project to address the 2015 Ocean Acidification Maine Commission report that wanted people to examine natural solutions for remediation of ocean acidification, and specifically to look at phytoremediation and co-cultivation of kelp with other shellfish. And at the beginning of this project, we created this conceptual diagram of the halo effect and sought funds to support research to determine the size and effectiveness or relative magnitude and variability of the theoretical halo effect. Next slide. So we proposed to do this work in Casco Bay, in Portland, Maine, because it was the perfect place to study impacts of coastal acidification. We have obviously the atmospheric carbon dioxide driven ocean acidification happening, but we also are subject to some fairly corrosive events after major rainfall and as a result of having a rather large watershed. In addition, Casco Bay here is home to more than 200 shellfish lease sites, so it is a very active aquaculture location. So next slide, please. Within Casco Bay, we worked at the ocean-approved sugar kelp farm site. It's the site of the first successful commercial kelp farm in the US. And so we were going to deploy our oceanographic instruments directly in this farm site, but we also sought to find a control location to measure comparative results. And to identify this location, we worked with Paul Dobbins, the owner of Ocean Approved, and Nick Record at Bigelow Labs to use an FVCOM circulation model that had been developed by Ernie True and validated by dye studies to determine the predominant tidal flow across the farm and identify a location that was relatively upstream from the farm. So you can see that second yellow star in the graph there is our control site. Next slide. Within the farm and at the control site, we deployed a set of oceanographic sensors that were measuring things like the partial pressure of carbon dioxide, seawater pH, oxygen content of the water, etc., and took discrete water samples every 2 weeks for calibration purposes to make sure the instruments were behaving properly. We also monitored the growth of the kelp on the farm. And as you can see, these operations are under rather cold conditions because the growing season in Maine runs through the winter months. So this was a burden of love, these kinds of measurements. Next slide, please. So I won't try to overwhelm you with too many graphs, but there are some really exciting results that we found early on that I wanted to show you. So along the x-axis of these graphs is time, and along the y-axis, in the top graph here is the seawater pH, and the bottom graph is the aragonite saturation state. It's kind of an indication of the calcification potential for organisms grown in that seawater. And in all of these graphs, the green lines or green dots represent the sensor data coming from inside the farm, and the black represents the sensor data coming from outside of the farm. And what you can see is that the seawater pH and saturation state is consistently higher within the farm than outside of the farm. And the red line going across the bottom graph represents the theoretical or purported threshold below which conditions are fairly corrosive to shellfish, acutely so. And you can see that within the kelp farm, I mean, Maine is very cold, so we get to these conditions every winter. But within the farm, you escape out of these conditions 3 weeks earlier than otherwise. And if you advance the slide 3 more times, please, Jesse, you see that we see these same results repeated year after year. And not only that, but as the kelp gets bigger later in the growing season and enters that exponential growth phase, the difference between inside and outside of the farm becomes more dramatic. Next slide, please. In year 2, we moved to include a mapping effort that used oceanographic sensors mounted on our small research vessel so that we could create a heat map of the CO2 conditions in and around the farm. And we drove our vessel in concentric circles around the farm, recording data as we went. And you could see that a gradient that corresponds with that predominant tidal flow coming from the upper northeast and down to the lower southwest sets up where, while the seawater CO2 content is higher outside of the farm, as it washes across the farm, the CO2 content decreases significantly. Next slide, please. Actually, I'll just mention, we did that mapping effort about 6 times in total and found that there's the halo effect is a bit of amoeba-like shape happening around the farm that moves and shifts with the tide. But we wanted to answer the crux question to this research, which was, what does this halo impact mean for shellfish, or in this case, blue mussel production in and around the farm? So we conducted a field transplant experiment deploying caged mussels taken from Bangs Island Mussel Farm, which was nearby in Casco Bay, and deployed these cages right in the farm site, and then at increasing distances away from the farm site, up that gradient that we identified in the last figure. Next slide, please. So after 2 months of this outplant experiment, we discovered that these blue mussels grown inside the farm had significantly denser and thicker shells using digital radiography and X-ray imagery to come up with this quantification. And as you move outside of the farm, even just over 100 meters away, that physiological impact of the halo effect is lost. Next slide, please. We had a very enterprising undergraduate student in the lab who developed a double pulley system to test the acute pressure and force thresholds of the shells that were grown inside and outside of the farm. And she used basic physics and dye-containing pressure paper to quantify those forces and acute pressures needed to break the shell. In essence, the dye paper turned a deeper color, the pressure paper turned a deeper color if more force was used to crack the shell. And we found that shells from mussels grown in the farm were much harder to break. Could you please click, Jesse? I think the video might show. Yes, this is really relevant to mussel farmers because during the size sorting process, you can lose a significant amount of that yield to shell cracking if the shells are too thin. And so even now, not 50 years from now, but today, this effect of thicker, denser shells could have a meaningful impact for shellfish farmers. Next slide, please. So this has been a very quick run through of a lot of research that has happened over the past 4 years, but we have some really exciting next steps that we're working towards now. Advance, please. First, we really want to determine the underlying mechanisms behind these co-culture benefits. Yes, we think that ameliorating the water quality has a role to play, but we're also curious about to what extent the detritus from the kelp farms can act as a food subsidy for the mussels grown nearby. And next, we're also curious about how these detrital subsidies may contribute to carbon burial in sediments below the farm, and in essence act to be part of the true carbon sequestration process as well. Next. We are really interested in testing this phytoremediation or halo concept at other sites with different hydrodynamics, because we think that the sheltered nature of Casco Bay played a key role here in the success of detecting the halo effect in this study. Next slide. We would also like to work with farmers to advise on operational design for co-culturing kelp and mussels that would both maximize the resilience of the mussels to ocean acidification, improve mussel yield, decrease time to market size, while also adding revenue from the kelp farming itself. And finally, we're really starting to lift off on a project to explore the potential for kelp to extract nitrogen from the water column and for ways to monetize that process. And there's a lot of funding support that we've received to support this work, and I just want to acknowledge all of those below, and then end with my last slide, advance please, about discussing how we're using these results and disseminating them. So we have 4 manuscripts in preparation by our new postdoc, Aurora Ricard, who's diving full head into these. And then if you just keep clicking, Jesse. We're also working with early adopter aquaculturists to design some of these cold cultivation techniques, and numerous PIs on these awards serve on the Maine Climate Council in our Science and Tech Subcommittee and are working to develop a blue carbon strategy for Maine. And I'm also advising through the Seaweed Fisheries Advisory Council of Maine and on the Seaweed Council. So we're working hard to get these results where they may be most useful. Thank you, everybody, for listening.
Thank you so much, Dr. Price. That's always a great presentation and so appreciated. I think we'll hold questions till the end, but also feel free to maybe type questions in the chat box, and we'll see how we can navigate that as well with our speakers. Lovely. All right, so moving over to a different location in Alaska, very happy to have Jacqueline Ramsey with the Al-Utik Pride Shellfish Hatchery joining us. Jacqueline, can you hear me okay?
Yeah, I can hear you fine. Can everyone hear me?
Oh, great. Yes, I can. All right, so we'll go ahead. Sorry, I'm just letting participants in. Go ahead and get your slides up and take it away. And thank you. I'll just say thank you, Jacqueline, for being here this morning. And it's really great to have you part of the OA Alliance family. As look forward to hearing your presentation.
Well, thank you, Jesse. Thanks for inviting me to present today for the Al-Utik Pride Shellfish Hatchery. We are, you're looking now at Resurrection Bay. And on a day like today, it actually does look like that. We do occasionally have beautiful days like this. So probably right now there's all kinds of photography going on in Seward. So I'm Jacqueline Ramsey, and we can advance to the next slide if we can, Jesse. The Al-Utik Pride Shellfish Hatchery is the only shellfish hatchery in Alaska that produces all of these different species. We dabble in oysters, gooey ducks, basket cockles, Pacific razor clams, little necks, purple hinge rock scallops have come through our door before too. Blue king crab, excuse me, and red king crab, which is a big production here every year. I think we've just had our last production year for red king crab. But we've done that now for going on 10 years. Is that right, Jeff? California sea cucumber, we have pinto abalone and butter clams. We can go to the next slide, Jesse. So a number of years ago, just to kind of jump from the hatchery and its production into OA, when we found out what was going on in the lower 48 along the West Coast with oysters, we were approached to find out what was going on in the hatchery. Next slide, Jesse, please. And with the, and again, one more advance. We were approached by oceanographers, Wiley Evans at Hackeye, Jeremy Mathis, who was at UAF at the time, and Dr. Burke Hales, to find out what we were finding in our hatchery, and we didn't know anything about ocean acidification at the time. So we collaborated with them in order to put in a percolator. Next slide, Jesse, please. And here's Burke down on your lower left and Wiley on your lower right. And the system that we currently have in our hatchery, the version percolator that we have now in the middle there. So we've been doing, we installed this for continuous monitoring originally, and we have been, we can advance the next slide, Jesse. We've been doing continuous monitoring now since 2013. And this is a slide from Dr. Evans. I wanted to just have you focus on the upper left graph with the two circles. Those are the times of the year that we find corrosivity in our waters. And that was an important, that was some important data, finding out that we do have two specific periods of time, at least in this time series, that we do have corrosivity in our water. Next slide, Jesse. As I mentioned, our continuous monitoring has been going on since 2013. We have two data portals that our real-time data goes out to, one at aos.org and one at ipaccoa.org. And you can find out what our data is in real time through those data portals. Next slide. So after we built the capacity for water monitoring, we received BIA grants to conduct a sampling program here in South Central Alaska, where we have native villages with science monitors that take water samples for us. With the percolator, we can do discrete sampling, and so we're expanding our monitoring efforts on the Kenai Peninsula, and we've also expanded to begin to monitor on the North Slope. Next slide, please. So this is a shot just showing our ability to do the discrete samples on our wet lab bench here. You can see my hand loading a sample for discrete monitoring, and the screenshot above that shows the percolator's computer screen interface. Next slide. So this is a shot of our community sampler in Seldovia. This is Stephen Payton, and he's sampling off one of the docks in the wintertime there. This is the sampling kit, the low-cost sampling kit that we devised with Wiley Evans and Burt Hales, and through the protocols of PMEL and HACI and OSU, we put this water kit together. It contains the protocols, bottles, the caps, the cappers, paint pens for marking the bottles, sample logs and fixatives, and thermometers for taking the in-situ temperature of the water sample as it's taken. These kits are packed in a rugged action packer that can go through the US mail because the fixative is in such low quantities in the samples. Next slide. This is a map showing where we are and some of our villages with respect to Anchorage there in the top. There's the Kenai Peninsula. And what we have been trying to do is get more sampling sites so that we can find out what the signature of OA is in these very different places. We already know that OA in the open ocean is much different than at our sites. In Seward, we don't have upwelling that we need to deal with. What we have is seasonal freshets and glacier melt that uh, um, provide corrosive waters at different times of the year. And we're not finding identical signatures anywhere at any of our other villages. They're all a little bit different. So near shore oceanography is much noisier. Next slide. So just to kind of wrap up a little bit, we have the resources here at the Allutic Pride to produce many different shellfish species. And being able to have the resources of the Berkelator and the hatchery personnel here, we're able to provide the ability to do experiments on a lot of different species and larvae on OA. We have conducted in the past butterclam research with Dr. Kelly's lab at UAF. We are also moving into kelp production and OA mitigation research. We have some things on the horizon for that, if any of those targets hit. We also are in collaboration right now with Dr. Hills and the Ocean Foundation, Alexis Valerie Orton, for real-time PCO2 monitoring for hatcheries. I'll let Alexis talk more about that. Hopefully on the horizon, we'll be broadening our research into harmful algal blooms and shellfish tissue testing. And that's what I have for today. And thank you to our supporters and our sponsors.
Wonderful. Thank you so much, Jacqueline. That was a great presentation and really cool to see the maps and different areas around the region, as well as thinking about sort of what's on the horizon for trying on some of these adaptation pilots. So really look forward to staying in touch with you about how that's going. And again, thank you for joining us today. It really means a lot.
Well, thank you very much for having us, and really want to thank again. We couldn't do this without Wiley Evans at Hackeye and Burke Hills at OSU, excuse me.
Great. Well, we'll get to hear from some of those folks as we go through the morning or afternoon here. So that sounds great, and was a good queue up. Jacqueline, you mentioned Alexis, our next speaker from Ocean Foundation. Alexis, can you hear me okay?
Yes, I can. Can you hear me?
I can. So we're going to go ahead and let me just quickly check my chat. Great. So Alexis is going to talk a little bit more about the Ocean Foundation's work. Ocean Foundation's work to build capacity for monitoring really all over the world, and might touch on some of those kits that Jacqueline mentioned that will be more specific for hatcheries internationally and throughout even North America. But I think we'll focus mostly on these kits that they have been deploying for the last several years now that are increasing monitoring locations all over the world. So I really look forward to hearing from you, Alexis, and I will go ahead and just be quiet and turn it over.
Great. Thanks, Jessie. So we talked about we're talking about innovation today, and I think a lot of times people think about innovation as needing to create something new, but sometimes I think innovation in a way can just be about making things more equitable and distributed, and really trying to invest in people locally. And so that is a large goal of the program that I run, is trying to invest in local leaders and build equitable distributed capacity to respond to ocean acidification. Next slide. So the Ocean Foundation has been working on ocean acidification almost since its founding. We originally started off trying to just raise awareness about the issue within the philanthropic community, but we have really refined our strategy over the years, and now our goal is to help vulnerable human communities monitor changes in ocean chemistry, understand the risks to their social and economic health, and respond to the effects with good policy based on good science. Next slide. We work with a large number of partners, and we can't even fit all of the logos on here, but I just want to emphasize that we are not doing this work alone by any means. So everything that you'll see from here is because of all of the people that we work with. Next slide. So we've talked a little bit about, as probably most people on this call have been on other OA Alliance calls, but ocean acidification is inherently a global problem. We know that it is happening in different ways in different places, so it's not the same here in Seattle as it is in Seward, Alaska, or in, New Zealand. And we can't, the problem is right now we don't have a global data set to understand this issue. So I have just a couple figures illustrating that. This is a map that's where the country size is weighted by the number of ocean science publications. This I've seen it a few times, but I got this figure from the IOC UNESCO Ocean Decade report. And so you can see that there are places like the continent of Africa, right, is almost non-existent on this map, because there's just not very much ocean science being published in that region. The next slide is focusing specifically on ocean acidification. This is something that the IAEA, International Atomic Energy Agency's Ocean Acidification International Coordination Center put out. Note that this refers just to first authors, but you can still see that most papers are being published in a small subset of countries, and not necessarily the places that are most vulnerable to ocean acidification. So this is sort of the challenge. Next slide. So we at the Ocean Foundation, we're trying to figure out how can we make OA monitoring more accessible and equitable. And we heard time and again that the systems that were being used, while excellent in a lot of places like the US and Europe, just were too expensive, very difficult to maintain, required kind of constant calibration, and just weren't really fit for use in places that might be a bit more remote or not have engineers on site who can fix electronics. And so we designed together with our partners something that we nicknamed Go On in a Box. Go On is the Global Ocean Acidification Observing Network. So I'm going to walk you through kind of what's in the box and why we designed it that way. So step one to using our kit is to collect water samples. And there's actually, a lot of things you have to consider when you do this. You want to make sure that you're preserving the sample in a bottle that's not going to react with the water and change the chemistry. You even need to worry about the type of plastic tubing you use to collect that water. So we focus on helping people get a good supply of those bottles that you need and the sampling materials, as well as the poison that you put in the water to stop growth, basically. Next slide. The next step is to analyze the pH. We use this thing called a spectrophotometer. There's a picture of it if you're not familiar with this type of device, but we make all sorts of little decisions when we design these kits to make them as user-friendly as possible. And for example, we chose this spectrophotometer because it has a screen built in that gives you the readings and is sort of designed for kind of rugged environments where there might be high humidity or other things, whereas another spectrophotometer might require a computer interface to be plugged into. We also provide all the reagents and everything you need to run these analyses. Next slide. We also provide equipment to analyze the alkalinity of the sample, and this is a system that was designed by Andrew Dixon and then tested by Chris Sabine. And all it really did is said, let's make this a manual thing. Instead of having it in a black box where you push a button, let's just take it apart into a bunch of little parts because it makes it way cheaper, and it also makes it easier to fix if something breaks, because it might just be one small thing that you can order for $100 rather than these all-in-one systems that cost $20,000 and are extremely hard to repair. Next slide. We also recognize the need to measure the chemistry in situ, and so we've worked to try and use kind of new technologies that, for example, the sensor that you see in the water is the iSAMI sensor, which won the X Prize competition for a pH sensor. For affordability and precision. And we also use a low cost CTD. So we try and use things that are small, that are less likely to be noticed and stolen in different waters, and that don't require calibration. So that you, if you're in a really remote location, don't have to worry about sending something back to the manufacturer once a year. Next slide, please. And then we also provide a computer and all the software required to do the calculations needed to process the data. Next slide, please. So these kits cost about 20,000 US dollars for everything. And in comparison, you know, some of the systems that are being used, say here in Seattle, are upwards of 300,000. And they're wonderful systems. It's just that it's not going to work in some places. So we've shipped 17 of these kits to 16 countries, and in a lot of places, this is the first OA monitoring that's happening. We do a lot of training alongside this, so it's not just that we're sending equipment, but we work with the scientists receiving this. One of them's on this call. I'll give a shout out to Marcia in Jamaica. And we do in-person trainings, and we try and do remote training as well. Next slide, please. I want to note that that's not the entirety of our program. There's a lot of other aspects to it. One big one is policy, and we work to train legislators on the best legal frameworks to address ocean acidification. So this is an example of a resource that we've developed for policymakers. It's basically an assessment of every existing piece of legislation or legal framework in the world that has been passed to address ocean acidification, and an assessment of sort of what the purpose of it was, as well as links to the actual legal language. You can use it as a template. And we hope to keep this updated every year. Next slide, please. So in conclusion, what we're really trying to do is lower the barriers to addressing OA. We don't think that the solution should be only available to those with power and money, right? This is a global problem that, like most global problems, is harming those in lower income places more. And so, you know, we're really trying to think about free things we can give. We never charge money to our participants. Everything we pay for, all their travel, all of their equipment and everything, because we're really trying, and we provide stipends, support to try and give people a little bit of seed funding to get going, just to lower as many barriers as we can. So we've done a number of things, and as Jacqueline mentioned, one of our newer projects is to try and kind of take what Burke and Wiley developed with the Berkolator and say, you know, what can we do that has a different purpose, is not for high quality monitoring, but is just for shellfish resilience? And how can we design a really low cost, simple to use system that will make it easier for shellfish hatcheries around the world to adapt to ocean acidification? So with that, I hope you got a snapshot of our programming and sort of our goal with it, and I'll turn it back to Jessie.
Thank you so much, Alexis. Excellent presentation. And it's just an honor to be working with you guys and to be learning from all the work that you have been involved in for so many years, and really appreciate your time today. And And I think with that, just let folks know that we have heard a lot about Dr. Wiley Evans throughout the presentation. So it's my distinct pleasure to be able to introduce him as our next speaker and talking a little bit more about what the Hakai Institute has been up to for several years relevant to ocean acidification, but with a particular focus on thinking about the utility of public-private partnerships and passenger ferries, which are obviously very present and part of our Pacific Northwest experience over here in North America, and how to utilize those cruise vessels to get more monitoring and cover more space. So I'll turn it over to Dr. Evans, and then also note that he'll be our last presenter right before closing remarks and a presentation, second to last, I should say, before closing remarks from Peace Boat and Emily McGlone. So Wiley, can you hear me okay?
I can. Thank you, Jesse.
Great. Just making sure I'm getting folks in the meeting here. Okay. So I'll go ahead and get your slides up and turn it over.
Okay. While you're bringing it up, hello, everybody. Thank you for the opportunity to talk to you today about a project we're really excited about. I want to highlight that while I'm going to be talking about using vessels of opportunity to collect data for monitoring ocean conditions, it's not a new approach. This approach has been utilized for a number of decades now, but our project with the Alaska ferry system is new, and we're collecting data in places that we haven't had any measurements before. And so what I want to highlight here is that this is a multi-institutional and international project. I have all the agencies' logos on the bottom of this slide, but it's important for me to point out that they're agencies that represent both Canada and the United States. And the idea is that we outfitted this particular ship, which is the motor vessel Columbia, one of the Alaska Marine Highway ferries that runs between Bellingham, Washington, and Southeast Alaska every week. We outfitted it with a CO2 system to monitor surface conditions while the ship was underway. Next slide, please. This was not a small feat. In fact, it took close to 2 years to get approvals, funding, engineering, do the shipyard work, and then finally be underway with data collection in late 2017. So it was a long process. But just to sort of walk you through how things are configured on the vessel, we use a general Oceanics 8050 CO2 system, which is unfortunately one of the units that Alexis was pointing out is kind of on the higher end of cost. But we have this system installed on the vessel in the car deck, and we pull water from the bow thruster cavity, which the photo on the left here, that cavity in the bow, that's where the bow thruster propeller is. On the top of that cavity, we have a seawater intake, and seawater comes into the vessel there. We make a temperature measurement immediately. Water is then diverted up to the car deck where the CO2 system is, make another temperature measurement, as well as the salinity, dissolved oxygen, and the atmospheric and seawater PCO2. In 2019, we teamed up with Yui Takashita, who's from the Monterey Bay Aquarium Research Institute, and Yui installed his biogeochemical surface monitoring system, which is a custom MBARI system that measures pH and nitrate. And so we have a broad suite of measurements that are being made every 2 minutes on this vessel as the ship's underway. I want to point out that this type of private-public collaboration isn't just the use of the platform, but involves people, and that's an important aspect for the success of the project. In the picture on the top right is Christy Harrington, who's the environmental program specialist from the state of Alaska Department of Transportation. And Christy is probably the MVP with this project. She has done an amazing job ensuring that the instrumentation on the vessel runs flawlessly. We have things set up so that they can run autonomously, but every time the ship is in port in Ketchikan, Christy goes on board and makes sure that the system is running well, and that's ensured our success. And so I really want to highlight that role for this type of private-public collaboration, that it's not just the use of the platform, but it involves people. Next slide, please. So what are we learning? We're learning a lot about how the CO2 content in surface water varies in space and time along the transit. And I want to take a few minutes and just orient you to this figure. The x-axis is the longitude, the y-axis is the latitude, and the z-axis is time. And so you're going from November 2017 on the left side of the figure to September 2019 on the right side of the figure. And each squiggle, each line is one transit. There's 135 transits over the 2 years that we've collected data in this figure. And the color of the data is the seawater CO2 content. And so warm colors are seawater CO2 conditions that are above the current atmospheric level, with red being nearly 2 times the current atmospheric level. And you can see the winter months, the surface CO2 is always generally higher than the atmospheric CO2. And there's areas where it's really high, where it's red. These are tidally mixed zones. The blue color are conditions where the CO2 gets drawn down to below the atmospheric CO2 level. And that happens typically starting in the spring when the phytoplankton communities begin to grow and photosynthesize and utilize CO2 in the water column, drawing it down. And so what we're seeing is the phytoplankton blooms in the spring and summer in those areas in blue. And so you can tell there's a big seasonal signal, but that seasonal signal varies along the coast with these regions that are breaking up, the places in blue that are these tidal mix zones where CO2 is maintained at a high level through the year. It's a pretty complicated and excitingly interesting picture. And there's also subseasonal and interannual variability that we're beginning to understand. And for example, the timing of that spring bloom, it doesn't always initiate at the same time each year. In fact, between year 1 and year 2, there was a few weeks difference in when the spring bloom started. And how it evolves in a particular area is also different in terms of its intensity and the spatial extent of that CO2 drawdown. And so we're not just considering CO2 content from the data collected from the ferry, we're also able to resolve the other CO2 system parameters using regional relationships that we validate with samples that we're collecting on board the vessel during what we call ride-along cruises. And one of the parameters that is of specific interest is this saturation state term that Nicole introduced earlier in this session. Next slide, please. And so the data from the ferry really amplifies our observing portfolio in this region from Southeast Alaska to across the BC coast. What I'm showing here are the time series locations in this area. On the left are the time series sites in Alaska. Starting on the top is the Berkelator time series from the Sitka tribe of Alaska's Environmental Research Lab. The next panel down is a CO2 mooring that was maintained by the Ocean Acidification Research Center at the University of Alaska, Fairbanks. And then the final panel on the left side is another Berkelator record from a shellfish aquaculture facility in Ketchikan known as Oceans, Alaska. On the right are the BC time series locations, the top being a CO2 mooring that we maintain on the central coast of BC. The next panel down is from our field station here in the northern Salish Sea, is another Berkelator record. And then finally, the last panel on the right is another Berkelator record at a shellfish aquaculture facility in Baines Sound here in the Salish Sea. And so you can see there's seasonality in all these records. There's a lot of variability, but what I want you to pay close attention to is the number on the top of each panel. That is a metric called the coefficient of variation. It tells us how variable that time series is. And what I'm showing in the middle panel is that same metric from all the ferry data. And so you can see while these time series are hugely valuable and providing important information for their specific locations, the ferry data is really filling in the gaps between the time series and painting a picture of how that variability changes in space. For instance, in Sitka, the ferry data coefficient of variation for this term matches what the time series is for the Sitka tribe of Alaska, but just north of there in the mixing zone in Sturgis Passage, it's quite different. And so that picture of the spatial variability is really important, and that's what the ferry data is helping us to understand. Next slide, please. And we can use this information on the variability that we're understanding from the ferry data to help guide our future observing strategies. The panel on the left shows the standard deviation of PCO2 from the ferry data. And from this term, we can estimate another metric called the time of detection. And this is the time in which it would take for a long-term trend to emerge from a noisy data set. And so in places where conditions are not that variable, it takes less time for a long-term trend to emerge from that noisy data set. And so you can see, like between Vancouver Island, for instance, and the mainland of BC, that area in blue, it's not a highly variable region, and the time of detection in that zone is very short. And so if our goal is to observe long-term changes in these dynamic coastal settings, it would be very helpful to target our observing efforts in places where the variability is low, so we have a chance to observe that long-term change in a much shorter amount of time. Next slide, please. And so finally, I want to point out that working on a ferry is a really excellent vehicle for outreach. It's not just a great platform to monitor data, to monitor ocean change. And we've had the opportunity to engage with class groups that have come on board and gotten tours and learned about ocean acidification. I've presented to riders on the ferry while on board collecting validation samples on the long transit from Washington up to Southeast Alaska. And we have a great media team that's generated a number of posters that are on board the ferry in various locations, as well as in the Alaska highway system terminals. And so it's an excellent platform to reach people and get more folks engaged with this problem that we're all focusing on, ocean acidification. With that, I'll turn it back to you, Jesse. Thank you.
Thank you so much, Dr. Evans. That's great. And it's cool to see those pictures at the end. Absolutely. So I think with that, I'm wondering if there's any, I think we have a little bit of time for questions, and I'll turn it over to our last presenter, who's our another co-host, and will give us some concluding remarks as we move towards the end of UN Oceans Week. But with that, any questions that folks have, and feel free to maybe type them in the chat box, or just unmute yourself and feel free to ask that way. That's fine too. Nicole, it seems like you have a question for Wiley. Go ahead.
Yeah, Wiley, thanks for that presentation. It was really great. I'm really curious in this ship of opportunity approach. A scientist at Bigelow has been doing that with some of the ferries going across the Gulf of Maine up to Canada and back. And in that effort, they're comparing what they're seeing with some satellite imagery data too. And I was wondering if you're taking that approach in the Pacific Northwest to look at the timing of phytoplankton blooms and the changes you're seeing in the ocean chemistry, or any of the satellite data that could be used to look at CO2. You could use some of those data to validate that bit of satellite information as well. It's just a really powerful data set you have there.
Yeah, thank you, Nicole. We've thought about satellite data in the context of the phytoplankton blooms a little bit. Specifically, there's an odd region in Queen Charlotte Strait where the CO2 maintains a very high level, and yet the satellite chlorophyll also is high. And so it's a bit perplexing what's happening there. And so I've sort of focused in on that region and looked at the satellite data. There's also a postdoc in Alaska working with some of these data and looking at 551 nanometer water leaving radiances, which is a metric of turbidity, to try and understand sort of the freshwater influences on the CO2 chemistry. But beyond that, not much else. In Alaska, it's very cloudy. And so that's a problem for some of the satellite products that we may typically use in other areas.
Thanks, guys. And I'm seeing a question about the first presentation from Karam in New Zealand about the report referenced about OA impacts on snapper. And the question was, is that published yet and publicly available? The answer is I don't know. I understand that some of the content in Cliff's presentation has not been yet published, so there is some sensitivity around sharing too broadly. But what I will do is follow up once that paper is made available, or Celeste, I'm even happy to connect you directly. So feel free to email me or I'll email you so we can talk more about that, but great question. Okay, so I think with that, and looking at our time, I'm very pleased to turn it over to Emily McGlone with Peace Boat, who has brought us all together for this UN Oceans Week as part of a larger collaborative of actors working to fill out events this week on this important platform. So thank you, Emily, and thank you to all of our speakers who are here today and all of the participants. It's just a wonderful community to be part of, and very grateful for your time. So Emily, can you hear me okay?
Yes, I can hear you fine, thank you.
Lovely. So what we're gonna do before Emily speaks, right, is share a short video about Peace Boat and then turn it over to you. Is that correct, Emily?
That's great, thank you.
Cool, okay, so I've got the video up here and we'll see that I can hopefully make it big for everybody, and we can also share the link in the chat box in case it doesn't feel, let's see here, in case it doesn't come through well for everybody. Let's see here. Hopefully it will. Oh. To everybody. Okay. So close that, and here we go. Turn the speakers up.
Okay, thank you so much, Jessie. And again, thank you to everyone who's participating in today's webinar. I think that gives you a little bit of an idea of our organization, Peace Boat, and our newest, most innovative project, the EcoShip. So just to introduce myself a little bit, my name is Emily McGlone, and I'm working as a director of Peace Boat US, which is an international organization based here in New York City, where I am calling in from today, but our headquarters is actually based in Tokyo, where Peace Boat was founded.
Apologies.
No worries. And if you want to scroll up, Jessie, just to the first slide there, that would be great. Thank you.
Of course, there we go.
Sure.
All right, let's try this again. Oh gosh, oh gosh.
That's okay. Great, thank you. So as you saw in the video, which is really exciting for us to have this rendering of the EcoShip, which we're working on right now as our main focus of Peace Boat as an organization, I think it just gives you an idea of the scope of what we hope to do with our newest boat, which will be featured here, sailing as a flagship for climate action and sustainable development goals. Next, please. And Peace Boat has been sailing around the world for more than 37 years. It was founded in 1983 in Japan, actually by university students who were looking to build bridges and cross-cultural communication and partnerships around the world. And they decided to take a ship from Japan to many other countries. And in that way, helping to understand the global issues that are at hand. And Peace Boat was nominated in 2008 for the Nobel Peace Prize, but we actually carry it now on board as part of our partnership with ICANN, which is an international campaign to abolish nuclear weapons. And we sail with the Nobel Peace Prize on the ship since 2017. So that's also really exciting for us as an organization. And you'll see a few facts here, but basically we are traveling on this ship promoting peace and sustainability with more than 1,000 participants or passengers on board, and we visit more than 100 ports of call every year. Next, please. These are a couple of examples of the global voyages that we take with Peace Boat. As I mentioned, our headquarters is in Japan, so our voyages always start from Yokohama Port on the east side of the map, and then you'll see like the red line that goes in northern hemisphere up into the Baltic region, and the green line that goes into the southern hemisphere, visiting South Africa, Namibia, Brazil, and Argentina, and always going through the fjords of Patagonia. So we try to kind of arrange our voyages depending on the socio-political situation in the world, depending on also the partners that we want to visit, and the global issues that we want to talk about on board the ship. Next, please. On board is where we organize all of our educational programs, and as you might have seen in the video also, we sail with the United Nations Sustainable Development Goals logo painted on the hull of the ship. We are organizing many different programs around the SDGs, and in particular, as we're sailing on a ship, of course, is SDG 14 for life below water. And in this photo, we're also featuring our Ocean and Climate Youth Ambassador program, where we bring young people from small island developing states on board the Peace Boat to talk about important issues like ocean degradation, climate change, and of course, ocean acidification is part of this education. Next, please. So as we're raising awareness on board, we bring guest speakers who come on board to give lectures and workshops between each country that we visit. For example, if we're flying a guest speaker in, we will bring them, if they're from India, we might fly them to Singapore and have them join the ship for that section of the voyage, speaking about the important issues related to that area. So we're always working to raise awareness and provide educational programs to enhance the experience of visiting different countries and to really get a global perspective on the issues at hand. Next, please. And then when we arrive in the countries that we visit, we have our programs and events in port. For example, on the top left, you might see doing a beach cleanup and helping to remove waste from coastal areas. On the top right was also recently in New York City for the United Nations World Oceans Day last year. We partnered with the UN to actually hold the main reception on board our ship. We had about 1,000 participants joining us, from United Nations delegates to civil society and youth, and it was really exciting. One of Peace Boat's great contributions, I think, to the United Nations is serving as kind of a bridge, using the ship as a neutral space, as a venue to host events and to provide partnership building spaces where we can really have these important conversations. And oftentimes civil society is not always involved in the United Nations events, but here we have an opportunity to raise awareness about the Sustainable Development Goals on a broader perspective. Okay, next please. Peace Boat always tries to promote socially and environmentally responsible tourism as we travel on board the ship. We really believe that travel in itself can be a tool for positive social and political change. So as we're traveling and raising awareness, we're also working with our local partner organizations, whether it be indigenous communities or women's groups or movements for environmental awareness and ocean conservation. Next, please. And that brings us to the eco-ship, because we really believe that in order to sail as a flagship for the Sustainable Development Goals, we would love to have a more sustainable cruise ship. Next, please. And as you saw in the video, we have solar panels and wind sails, which we will have on the roof of the ship, using all of the renewable energy that we can harness from being on the ocean every day with so much sun and wind that will support us in traveling. And we understand right now the maritime industry is, part of the cruise industry. It's still a very small part. but it's also a very conservative industry. And these kind of cruise ships don't exist today. So our goal is to be a game changer and to support the new regulations that are coming to meet the targets for 2030. And we know that the shipping sector must reduce its greenhouse gases by 50% before 2050. So we're hoping to set an example to really kind of set a paradigm shift for the entire maritime industry. Next, please. And you'll see from the video also, it was mentioned in the slides, but we're hoping to reduce our CO2 by 40%, a 50% reduce in energy load, and 20% reduce in propulsion energy. So doing as much as we can, harnessing the renewable energy, and also raising awareness about energy use on board. For example, having meters in the cabin so people can see when they turn off their lights or when they're using less energy, they can really think about their daily habits on board as well. Next, please. And with that, some of the daily activities that we'll be including on the EcoShip will be on the top left. You'll see a kind of kinetic energy dance floor where you can, have your dance classes, but you can also create energy. It's really exciting. And on the bottom slide, you'll also see our marine observation deck, which we're hoping to have more of a conservation effort and awareness raising about the oceans. We're working with partners like Sylvia Earle and Fabian Cousteau to really bring the designs and innovative techniques on board the ships that people can learn about marine conservation. And I was just going to say, I was so impressed with our previous speakers about some of these kits and testing capabilities that you have, as well as, for ocean acidification, pH balance, temperature, and also for CO2. So I would love to partner with you all on working together on our current ship, but also towards the EcoShip in the future. Next, please. We'll also have a botanical garden on board, a kind of a plant kingdom where we can recycle the gray water in a closed loop system and irrigate, the plants that will be inside the ship. Next, please. And this is just a little bit of a highlight for some of our partners that we're working with. Many of you probably know. And we're always looking for more partnerships with Peaceboat and EcoShip. We're a global organization, and we understand that, the private sector, leaders in the environmental field, as well as civil society and the United Nations really need to come together to make an effort and to set an example for the future generations and what we can do to reduce our CO2 in the oceans and really help to conserve this really precious ocean resource. So with that, just in closing, I'd like to say again, thank you to the OA Alliance and all of the partners, because this year, the United Nations World Oceans Day has a 2020 theme of innovation for a sustainable ocean. And with these partnerships, I think that really we can work towards these targets and, building these partnerships one by one to reach more public and raise more awareness together. So thank you so much. And I really look forward to working with all of you in the future.
Wonderful. Thank you so much, Emily. And I'll just echo what a pleasure it's been to work with you as such an accessible and friendly person and really, truly working on collaborations. So I'm probably thinking the same thing that our presenters are thinking. But Wiley, do you want to ask your question or relevant to, talk a little bit about 14.3 and how partnership, perhaps, we could convene a smaller group offline sometime later this year to think about how we could help meet some of these targets and indicators for 14.3. But do you want to say a little bit about your thoughts on that?
Sure. I was just thinking and getting really excited about the potential for underway instruments on EcoShip, and I just wanted to bring it up again. I know I brought it up before we started the webinar, but what we found is that in thinking about the process of installing instrumentation on a vessel, it's helpful to get that process going early rather than later, when a lot of the hardware and stuff on the is already set and you have to reconfigure things. It's often harder. So I just wanted to reiterate that, and I think there's a number of the indicators that could be targeted beyond just ocean acidity. But yes, thank you.
Yeah. Well, sorry, Emily. Go ahead if I interrupted your potential response.
Oh, no, that's okay. I was just going to say thank you so much. And we are really excited, honestly. We just became a member of the Ocean Acidification Alliance this year after we were participating in COP25, the United Nations Climate Change Conference in Madrid. And it was great to see so many partners coming together. And we realized that there's a lot more we can do on our current ship. And I didn't mention in the presentation, but just really briefly, we have the Peace Boat vessel, the Ocean Dream, which we've been traveling already on around the world. And we did just also acquire a new ship called the Zenith, which will be our second ship, which is the first time in Peace Boat history. So on that ship also, there's a lot of space for innovation and creativity on how to use that space. So I would welcome any ideas and love to partner with all of you. So thank you.
Great. Well, this is Jessie, and I'll just say again, a huge thank you to everyone who participated today, to our presenters, Emily, Wiley, Alexis, Jacqueline, Nicole, and Cliff. It was great to have you. I think you all did an excellent job building on these themes of innovative solutions, which I know can be a hard word thinking about climate change and particularly about ocean acidification, but I think everyone did such a great job, as Alexis well said, that a lot of that is getting a better understanding globally of what are the local changing conditions, and what are the priorities, what are the most vulnerable resources, whether they're economic, social, cultural resources, and how do we start thinking about deploying these pilots and these strategies to protect them or build better resilience or adaptation into the work. So really great presentations, appreciate it. As always, please feel free to reach out to me at Jessie, Jessie at oaalliance.org, or just go to our website and you'll find all of that information. With any questions, and perhaps we'll reconvene a little bit later this year to talk a little bit more about how OA Alliance can partner with EcoShip on some of these ideas that were raised. I think that's a great vision to start moving towards. So thank you to everyone. Happy UN World Oceans Week, and stay well, and really appreciate everyone's time.